That was a very interesting talk. Gave me a lot to think about. I think Stuart wanted a new model. And that he's got a new model.
I mean, I think that was an AI one to a blind market. The only veterinarian in the room, I've waited 20 years for the animal breeders to figure out there's underlying biology to these changes. And it's time, seriously, that we started looking at that. And I applaud you, Mark, for thinking outside the box. I think that's really what we have to do. And we have to think about different ways to use these data. I hadn't conceptualized the idea that you could throw RNA sequencing to understand gene expression. It's not something we think about, but think about that, going into a genetic evaluation where we have gene expression. Part of that is fantastic. Mark, I mean, clock, you should be a chicken guy. Cheap, okay? Remember my comments about the levers. If you think about where the cost of sequencing and where do you drive efficiencies into that, it's what can we do for library prep? What can we do for sequencing? What can we do for data management? Okay. I will tell you that I was mentioning this to Wade Schaefer a little bit earlier. The day is here that in fact the biggest cost in this entire process is picking up the sample at the lab. When Troy sends one hair sample, and somebody else sends one blood sample, and somebody else sends one TSU, and somebody else sends a semen sample, I've got to process all four of those things. I don't. But you have to process all four of them. You want to screw up and make inefficiencies in this system? Send me 4 different sample types for 4 different samples. It doesn't matter how many I put on the sequencer at that point, all of the efficiencies in the system are lost. So if you really want to drive costs out of this, you've got to start thinking about how are you going to take that from animal side to the end of the data management. And right now, the way that we collect samples and funnel them into labs is horribly inefficient. It's the single largest cost in the entire process and will be for a long period of time. So, if you want costs out of there, Mark, the other problem with the Cargill system was not that it didn't work. The problem is it got the sample on arrival. By the time that sample got processed and the data was available to make a management decision, the cattle had been in the pen for 2 weeks. And there was no way Cargill was going to disrupt that 300 head pen, drag them all back up and resort them. And so a lot of the value was lost because we didn't get the sample at the right time in the animal's life. And so if you want efficiencies, you do it, but you really have to think about when are you going to do it? How are you going to get that stuff in? So, I mean, just, as this was going on, Steve, a few thoughts were bubbling through my head, and I couldn't wait for this presentation, and thank you for listening to that. That's a great job.
Okay. Thank you, Stuart. I think kind of listening to some of these talks, kind of hearing storage is a problem, storage isn't a problem, computing is a problem, computing is not a problem. So we'll find out. But what I'd like to start is to have the panel introduce themselves and maybe get a first few thoughts in terms of what they're thinking about low pass sequencing.
Well, Victor Pedrosa with Neogen. I joined Neogen going to a year ago, and I'm originally from Brazil, was a professor there, joined Purdue University for a couple of years, and then now with Neogen. So I guess the good take-home message is that we are discussing low sequence data, and I guess everyone agrees that this is not anymore the next step, but the actual step that we need to have. Adding to what Stuart said today, I guess there is no way back of using that type of information. Genotyping was great, but right now we have better tool with almost the same price that we had in the past, like for genotyping. So why not using that information? So my first concern is how we're going to be using that information. But as we can see today, we have Mark, we have Cedric and others already thinking about that on how can we use that information, how can we model that, and more than that, what strategies we're going to be using with that information to succeed in the end. So I guess this is a very positive discussion today. So I'm very excited about it.
Hey everybody, I'm André Garcia. I'm a senior geneticist with the American Angus Association. I've been with the association for just 3 years now, and before that, I did my PhD and a postdoc at UGA with Daniela and Ignacy. And I think a lot of the talks today were really interesting, and I think there's a lot of information that we might get out of sequence data in the future. I think right now, what is interesting, we hear in all the conversations, right, how can we drive that from the research area and bring that into a practical application to make or create tools, right, that will either increase accuracy of our predictions or create new tools that will add value to our breeders who are at the end of the day investing in those resources. And at the same time, at the practical level, we might get it into this panel later on, right, but also needs to be feasible from a computational standpoint so that we can keep delivering solutions to our breeders, whether that's on the weekly genetic evaluations or that's the turnaround time for their samples when they submit the sample to the association for Neogen or whoever their provider is. So thank you for the opportunity to be in the panel.
Well, good afternoon, everyone. My name's Ryan Boldt, and I am the lead geneticist for International Genetic Solutions. I've been there, I guess, part-time for 6 months and then full-time for the last 6 months, and learned a lot today. And I've always appreciated coming to this conference and some of these things that maybe don't get exposed to quite as routinely, learning where a lot of folks are at. And I think, to me, there was things that were maybe question marks before that I think maybe some strategies were potentially presented today to kind of address those, which I think is helpful as we kind of decide. But looking forward to the discussion with the other panelists. And
Okay, I got a question. Start with the panel that Mark brought up, and I guess Stuart talking about chickens. It's kind of like that chicken and egg problem. In terms of driving down the cost, we need more animals in terms of being low-pass sequence. But we also need the cost of low-pass sequencing to go down to get the more animals. Do you have thoughts in terms of how we can kind of bridge that gap?
I guess I'll give some quick thoughts. I think that's always been a challenge, I would say, with genomics in general. I think as we think about different breed associations over time and maybe what I would consider adoption rates of genomic technology in general, I would say that that's probably been a bit all over the board, especially, different associations that we get to work with at IGS. And, I think with that may be something that as it becomes more cost competitive with SNP genotyping or the technology that we're using now that, makes that a lot easier transitioning over and like maybe as discussed a couple of times today, using, I would say, the output in a similar methodology that we're doing now. So I probably, I guess in my opinion, kind of see that as our first foray into using low-pass sequencing, that as we continue or move forward with that, probably continue to expand use and adoption of that.
Yeah, and I would say that I think to bridge that gap, right, it is a conundrum, right, because we need money to get started with the data to maybe show some solution at the end and maybe the technology picks up. I think research would be a great space to bridge that gap where we bring, smaller scale that data into the research component, try to come up with tools or or improve the tools that we have, or come up with new tools that could add value to those that are invested in the technology, right? And once we have that kind of pan out on the research stages, then you can start, kind of releasing that in a production mode, if you will. I think for sequence data now, it's been a challenge to show the benefits of using sequence data for genetic evaluations for just prediction accuracy's sake. And there was a lot of discussion about this already today, whether we're going to need new models or different approaches. Now, there could be other technology that comes out of sequence data, right, that we could develop in that research phase that could add tools to the toolboxes of the breeders that will justify additional cost or transition into a different technology.
I guess adding to that, and as you see today, we're probably going to need to have much more information for multi-breed, like different breeds coming in place that, for example, if the need to have the approach that Mark is proposing, we're going to not rely on Angus or Holstein, or we need to have a very spread information, and of course, low-pass sequence data provide us different target regions that we're not going to be looking just in Angus, for example, or other breeds, but also for small breeds that can have other causal mutations that we are not be looking nowadays. So this is also beneficial.
Thank you. I'd like to open the questions up to the, yes.
I'm sorry, Victor, simple, easy one. And you may or may not want to answer it, but everything that's getting sequenced now at Neogen, are you planning on keeping all those FASTQ files or not?
Right now, this is not a long term, like we're not planning to keep all that information. That information, we are much more offering the option that we can keep that, but you need to pay extra for that, or you can keep it yourself. So that's the idea. And of course, maybe having different strategies as you propose here will be beneficial for us and for customers that wants to like decrease that storage space for sure.
And to follow up on that, you will get access to really large amounts of sequence data to build a really nice reference. Is that the plan, or don't touch customer data?
Of course, we're gonna need to have the green light from customers to have that option. We're not gonna just go ahead and do it because it's not the initial idea, but of course, with the customers and breeders association understand that Neogen can build that, we're gonna be more than glad to do it.
Right, thanks.
Okay, I thought I saw somebody back there raised hand earlier. Okay, as Cedric was pointing out, there's going to be lots and lots of this data coming in. And that's valuable that somebody's paid for, that there's a need in terms of having a good reference that's available. Do you have ideas in terms of how we take that data in some sense in a fair and equitable way to build the reference population, reference backbones for that would be a benefit to the entire industry and kind of going across breed associations and producers? But it seems like a big challenge to me.
We'll keep some thoughts together and started. I think that in that discussion, right, in the data sharing coming from academia, right, when you're doing a PhD or a postdoc, or if you're working on academia, I think it's a very noble goal, right, when we want to pull all the resources together for a common goal. And we heard that earlier today too. I think for us, the challenge there becomes, I mean, when I'm talking about the Angus data, I'm talking about all the members, right? All the breeders that participate and are owners of the association database. And so that's what we also need to take into account, how that plays out into the practical point of view, right? What does that mean in reality? And so That's some consideration that we need to have when it gets down to it, kind of the data that we're working with, we're just allowed to play with the data and try to come up with new tools and improve the tools that our breeders have. But at the end of the day, right, we're handling their data and their investment in that database.
I guess kind of IGS view or thoughts on that. And I don't know that we've come up with a good plan on this. I'll be upfront, or maybe any plan. I guess we're going to be completely honest. You know, but the challenge there with our evaluation is we are going to need a multi-breed reference set. I mean, we're going to, and what does that look like? You know, who becomes responsible for figuring out the animals that go in there? You know, that's probably something we still need to consider and figure out. And if others in this room have suggestions on that, you know, definitely all ears on that. You know, I think some of, I guess, what we've tried to do, at least, or several associations that I've been involved with, have tried to do, at least with sequence data kind of so far, is trying to marry those with other projects and maybe try to kill multiple birds with one stone. So that, you know, may be a potential another avenue. You know, I think it's always challenging as we look across maybe size of different breeds that are part of our different genetic evaluations. I would assume likely we would have more representation of certain breeds other than others, and you know, definitely some breeds that contribute across all of the composite type animals. And so I think probably from a reference panel that maybe makes that a bit more challenging for us, but at the same time may offer an opportunity for that to be used fairly widely, depending on you know, what's all included in that.
I guess just adding some comment to that, like seizing the opportunity that we have CDCP represented here, I see as an institution that take advantage of having different breeds together, that they, for example, can develop a crossbreed genetic evaluation using the information that they already have inside that institution. So this is also a good benefit that even Angus, for example, can take advantage, because we have a lot of crossbred with Angus too. So why don't use that information that you already have in-house to proceed to that genetic evaluation for crossbred too? That's another advantage for sure.
So Steve, can I ask a hypothetical question? So
Why don't we give you the mic?
no, so what is your vision for this? So I see a public resource, which is a set of animals that are sequenced that are the reference database. It's held by the USDA. It's funded by targeted sequencing to create these animals. And so there's this kind of database, and then there's this people who are plugging into that to use that for imputing low-pass sequence, right? And that imputed low-pass sequence goes back to the user, whether that's the Angus Association or IGS or whoever, right? So I'm asking the question, as an industry, do you invest in the reference and put that in the hand of a public domain and have that as a common asset for the beef industry? Would that be useful? I think it would. I think it would be, it could be and should be quite helpful to improve the accuracy of imputation from low-pass sequence that everyone would benefit from. Am I making sense with that?
Well, it makes sense to me, but I'm a statistician.
I tell you, where I get fuzzy is where is that maintained? Mentioned USDA, but my ARS colleagues comment that, you know, once you stick something like that behind a firewall that nobody can access, right, that's problematic. we at UNL have increased security so much that it's hard to use our own data. Well, but I think so I look at the concept, I think the question is where.
Yeah, simplistically, Matt, I've always thought that that's on Amazon or, it's in the cloud someplace, right? And you kind of go in there and you can use it. You can't perturbate it, and you can't affect it, but you can go in and you can use it and I'm thinking of it specifically from the point of view of imputation, from low pass sequence. That's its sole objective. Cedric, your question about who owns the pass queue, who gets access to the imputed data, who stores that, that's a different question. I'm just asking, let's address the first part, which is how do we affect that? And so that would be my comment.
Now, on that, again, for you and maybe for everybody here, is it time to revisit the reference populations of yesterday's when we were doing extensive phenotyping, genotyping, and sequencing as a publicly funded thing? With the caveat that this time we don't stick it all in our pockets of whoever did it and publish a heck of a bit and then make it go away. With a clear mandate to put everything up there so that we have intensive phenotyping through and through again. We don't depend on having out data that is commercial, is private, is proprietary, we can't access for one reason or another, which makes sense, of course, for commercial data. But it's probably time to just do a full sequencing, RNA-seq for good measure, and all the metabolomics, proteomics, and genomics, and put the whole thing up in a public space where we can actually try and develop new methods. So that's, I think, probably where we should be going back to, as we did when we were trying to create the 50K predictions.
Okay, this got people going. Ignacio first, okay.
Yeah, perhaps I don't know how to make comments about multi-grid data. So I look at all of the papers on multi-grid analysis, and there are many analyses done, many efforts. And the story is, if you want to evaluate F1 or what is F1, you need to have the reference population of what you have. If you have just pure breadth evaluations of crosses do not work. And then why is that? So we can talk about the hypothesis why it works. You need to have references of the right crossbreed type, and then things somehow work. So that's why, for example, it works so well in New Zealand for dairy, because there is an excellent example of why application. So crossbreed application works very well because they have so many crosses. They have more crosses, more at the same time than pure line. But if you look at other crosses, then it doesn't work.
I can talk loud. Well, it's also getting recorded.
So along Stuart's point, and I said this earlier, think where you want to be in 10 years. You don't necessarily have to know how to get there, but if you figure out what your goal is, and this is going to be a really easy question for me as an academic to ask, but it seems to me that the CDCB model has been very successful. Why haven't you done that for beef cattle? And if you decide to go that route, why don't you merge with dairy cattle? Because I still believe cows are cows, and you're going to be able to learn a whole lot more from all cows as opposed to sticking to individual breeds.
Okay. I feel like I need to give the panel an opportunity to weigh into all of this. What's that? Oh, that's right, Mark. The panel may never get the microphone back.
So in response to Cedric's question, I would argue, Cedric, that that reference population is the germplasm evaluation population in U.S. mark.
So
Yeah,
where do I download it from?
I'm sorry,
Where do we download it from?
it's not available yet, but it will be. We're under the mandate to get that done, so we're trying to figure out how to do it. So But we've already paid for the animals and for the phenotypes and for the sequence. And, you know, we need somebody else to go out and find the money to do the RNA-seq and epigenomics and the metagenomics and all those things. And I see all these grant funded projects that go out and find a few animals that haven't been measured for anything, aren't related to anybody, and they spend all this money on them, and it just doesn't make sense to me.
Agree.
So, in principle, it will be available. Can't go download it today. But, that'd be my response.
Give a
Maybe
timeline.
this is an idea for a joint project, to start a conversation for a joint project, like a big one, to start to.
Should start becoming available.
Yeah, but it looks like it. Yeah, just saying that, sorry. Maybe this is an opportunity of a joint project with different institutions and not talking just academia side, but also industry side, that we can all benefit on that, and bringing, again, different breeds, different institutions that we can download the information, and all gonna be, not only the institutions will be benefit with that, but primarily the farmers. That's the main idea here, right?
Yeah, I want to make a comment. I think Matt brings up a big point around, you know, when we're talking about the data sharing and the data security, right? Even within our own organization, right? IS makes, or IT makes it very hard for us to work with the data sometimes, because it's all, you know, and it has to be, right? I'm glad that we have those people, because you can't put me in charge of data security by any means. And so we need that, but I think that's also an important consideration, right? And of course, in our context, right, that data belongs to the breeders, so we need to make sure that data security is also taken care of when we think about that. From the perspective of the imputation and speaking to the low-pass sequencing part, right, I think maybe from the three of us, I maybe have the easier part of the job, because we have, you know, we're working only with the single breed population, so that makes everything real easy, right? To mention Ignacio's point on multi-breed evaluations and imputation for the low pass. So I'll be able to the other to comment on that one.
Try to follow that up. You know, I like Mark's comments of the germplasm evaluation group maybe being that reference panel, because that does encompass you know, a lot of the different breeds that we interact with. And you know, there's obviously been considerable effort to build those resources there. I guess maybe a question for you guys, or maybe the group in general. I think when we first started looking at genomic prediction, the thought was to go back in time as far as we could with animals. As far as reference population, is this something that we will have to be kind of continually trying to update over time? And what, as far as resources, would you guys kind of foresee continuing to do that? Is that GPE, you know, just as new animals are added to GPE, for example, if we're using that, or what are your guys' kind of thoughts on that?
Yes, I guess the answer is yes. We're gonna need to keep tracking on that, especially because we have recombination across different generations. So how, like in the end, as we have genotype right now, like if you don't keep tracking and inserting phenotype information, for example, how we can balance and keep updating that information that will kind of spread across different generations. So I guess the simple answer is yes.
Yeah, and I'm not sure, like, if we heard this morning, right, how with the low pass, right, eventually you update your imputation pipeline and you improve, and then genotypes change. I think we saw that, you know, with GBLUP or single step, those models are very robust to changes on maybe the SNPs in the evaluation, but then in the future, if you're thinking about different applications, right, or if you're talking about genetic conditions, or if you're talking about parentage assignments, I think that becomes really important, right? Because if you get a different call, once you update the imputation pipeline on the low pass, then we need to explain to the breeder why the pedigree changed, or or whatever other tools that we have that will be dependent on that, right? That will be that becomes a very important change, other than PPD changes.
Yes.
I have a quick question. We've really stressed the importance of a good reference population to encompass all the breeds that, really we want to include in the low pass imputation. Angus is not, you guys are going to have a problem getting animals into that reference population, but smaller breeds, ones that are maybe not sampled as much, not as many animals, how do we account for including those animals to make sure that we're not kind of overfitting them and they're just becoming part of the need of the other animals in the reference population? What do you guys see as like a possible solution for that?
Maybe answer that question is what Cedric is proposing. Like, isn't that time right now that we can come up with a new updated reference population that will include different breeds? So I guess, and right now we're not talking about millions and millions that as we talking about that like 20 years ago, like it's much less now. So I guess this is a great opportunity of doing that.
Can I ask you another question? So does the CDCB still require the AI studs to send semen on every young sire that's evaluated, whether or not they make it into the lineup?
No, currently we don't receive sample.
Okay. That used to be, I think, the CDDR, the Cooperative Dairy DNA Repository, where you were required to submit sample. How come that stopped?
We're still participating in the CDDR, and that is a totally separate agreement. That CDDR is kind of NAB regulated.
Okay. Okay.
But again, it's a totally separate.
So, I mean, is that something that the beef industry should be doing? Is, bowls that go into AI, you grab 5 straws, put it in the deep freeze, and at some point, if that becomes an important animal in the pedigree, you sequence it. What's the cost of sequencing now? What's the cost of sequencing? What? $200. 30x. 30x coverage, is a few $100. It's cheap as Porsche. I mean, that's the point, is that, you need to be thinking about it from that kind of perspective. What would it cost you, Mark, to, how many bulls would you want sequenced a year? 1,000?
I would hope that low passes used sufficiently, that the breeders are already having a genotype of low pass before they're using it.
Yeah.
And particularly if it's not selection, I don't see any reason why they wouldn't be. So it seems to me like perhaps an effort that's not really all that necessary, and maybe better to have places.
You're the smartest guy in the room here, as far as I'm concerned. So I won't question that comment, but, my point is that if you want to do that, it's a relatively low cost. It's pretty cheap to sequence animals, to put them into your reference population. I just think that's kind of a no-brainer. And putting in place a process to have those samples procured every year and then do them as required, I think it's something that the Beef Improvement Federation should take on as a responsibility.
Can I make a comment there? And I think from a research perspective, right, and we're talking about sequence data and to get started with the research and gather some data. And so we've been putting in some efforts, right, to maybe acquire some of the sequence data and kind of build that population. But I think we cannot downplay the cost, right? If we put this into a production perspective and think about our breeders, right? A couple $100 may not seem like a lot for a research project or a research budget on a few animals, but on the day-to-day applications of our breeders that we already request them to record these many phenotypes, to register the animals, to participate in the programs, and then increase the cost of the genotyping, right? So I don't think the cost is trivial. I think from today's discussions, I think it's kind of clear that the cost is still a big barrier of entry for that, right? And then of course, all the development of new applications are improving the tools that we currently have that will be extra deliverables for those users of those tools. I don't know if Ryan or Victor would have other thoughts on that.
Well, I guess it's fair enough, but again, maybe some consortium, maybe some joint effort can bring some differentiations to other breeds too. I completely agree with you, André, that it is of course a concern in terms of Angus associated are investing on that for a long time. So it's not just giving that information for others, but of course, we have other opportunities of bringing money to the table and bring some different animals that we can share some information. That'll be great.
Yes. I don't know the answer I'm asking. I wonder if we're thinking about the costing of this all wrong. Because we're thinking about a single use case for a seed stock producer to increase the accuracy of EPV. But if this penetrates the commercial industry, like get low pass on millions of commercial animals, now there's multiple use cases for those data. The data flow into genetic evaluation to increase EPV accuracy. It can also be used downstream for management decisions. So all of a sudden now that value is increased because there's multiple end users, and you would think that the costing could then be shared across the multiple users. So I'm just questioning if the whole notion of it being too expensive is really even the right question to ask right now. Now, all that said, I'd like the cost to continue to go down, right? But I'm just not confident we're thinking about it the right way. We're trying to take a new technology and put it in the same box we've always worked in. I just don't think that's appropriate. But I'd be happy to take arguments now.
Yeah, and I guess I'll start and then I'll share the mic too. I think, Matt, that's a great point, right? We need to be able to extract more use out of that data, right? And for that matter, I think even the medium genotypes that we have sitting on the database today, right? We can come up with different rules or different solutions for the breeders, right? And I think for sure that's a valid point. My challenge to the argument is we need to deliver something for them. And of course, I always go back to the genetic evaluation because that's what we're delivering right now, right? But I think your point is very valuable that we need to be able to extract more value out of that data, right? And then kind of bring new deliverables, whether that is for seed stock or commercial industry, that will help us share the cost for sure.
So, and I'm sorry to comment, that's not be gone tomorrow. And but what Matt says is absolutely true. Okay, so when you put something on a flow cell on a sequencer, it doesn't matter whether it's a seed stock animal or a commercial animal or a feed. And maybe I could put some horse stuff on there with some cattle stuff. I don't know if you could do that. Somebody who's smarter than I can figure that out. But flow cells and sequencing technology are not the same as chips, right? We have a specific chip for Angus, and we have a specific chip for this, we have a specific chip for that. And you're always boxed in by the dimensionality. Have I got, I can only put 196 samples on at a time. When I'm on a flow cell, it doesn't matter whether it's a high value sire or a commercial heifer or a feedlot steer. It just fills space on a flow cell. And so that's where you really need to start thinking about that, Matt. It's just filling flow cells. And it doesn't make any difference, what goes on there. And that should be your mantra, which is how many samples can you get on there as efficiently as possible? Because that will be the primary driver in cost. And if you can do that, then, you know, all of this becomes very, very easy and very trivial.
One thing I'm kind of wondering, way back when, we always talked about P being bigger than N. Now those have changed around. Now we're switching them back and forth again. I'm wondering in terms of if our definition of what low-pass sequencing is either becomes different or less relevant as the technology advances, not in the next 5 years, but certainly in the next 10 years. A lot of stuff is for DreamWorks 10 years ago and there. So not sure. As a statistician, it's all chemistry. It's perfectly cheap for me because nobody's asking to pay for anything. But if anybody has any thoughts along those lines?
I guess going back 15 years ago, the discussion was pretty much the same. Like, what is the cost of 50K? How we can storage 50K? How we can use that information for genetic purpose? So 15 years now, we're gonna have the same challenge, but I'm pretty sure that in 15 years by now, we're gonna say, hey, what are the next challenges? Mark point out a very good, interesting point today that how we can start to use biological information to bring to the table more accuracy, more reliable EBV, things like that. So I guess probably this is the next challenge for us. Right now, I don't see any more sequence data as a challenge.
I think that's, Victor, you made some great points. I think, to me, and that's why I pointed before, like, how do we solve the conundrum of, do we increase the volume to drive the price, or the price drives the volume, right? That's why I mentioned research, because I think right now we're going through those same phases where we're gathering some of the data and we're starting to come up with the research. And so, to me, that's how the research plays a big role in there, because then we're going to be able to demonstrate and show, here's the value of sequence data, right? And maybe like Matt's point, right now, maybe looking at increasing prediction accuracy is not the way to go, right? I think we've had some papers with different, Ignacy mentioned single step, GBLUP, base R, base C, base RC, right? We haven't seen in Cedric's presentation covered a lot of that too, but there may be other uses that we can start investigating, right, for the value of that data. And so I think that's where that leaves still, and we're still to that phase. So of course we don't want to be blind to the benefits, right, and shoot ourselves in the foot and say, 10 years from now, what's it going to be? I think what the research is what kind of going to guide us to get there.
Yeah, I don't know that I have too much to add, but I think probably the marketing of it is going to be different than what we did with SNP genotyping, because we looked a lot at increases in prediction accuracy as kind of our selling point for SNP chips and incorporating that into genetic evaluations. And like you said, Andre, we're maybe not seeing those similar increases that we saw before. And so maybe part of that is thinking about what are those other use cases, or where can we create more value from that additional information that's not as much, well, we're going to make your EPD 25% more accurate, or, for example.
Yeah, maybe this is more of a comment. One of the challenges, I think, that from a modeling perspective, Mark went through, we're talking about in terms of bringing in all of that biological information, talk about imputation, doing that at different levels. One thing it seems to introduce more than what we had in the past is different amounts of uncertainty as we're going through, and how we're going to incorporate that into the model. So if we're looking in terms of incorporating biological annotation information, is the I think one of the challenges is some of that information is good. Some of it's probably very good. Some of it's maybe not so much, but it might still be useful. In terms of the modeling? Are we just kind of hoping it all washes out, or are we incorporating that directly into the model? I think that's kind of one of the big challenges going forward, is in terms of incorporating the uncertainty directly into the models, as opposed to, I think right now we can't just say, hope it goes away.
That's certainly a great point, Steve. And so one of the things I didn't address that I glossed over is that this imputation of the omics data is a bit different than imputation of genotypes, in that when imputation of genotypes works well, and it's about as accurate as the real data, not completely, but it's pretty close. And clearly these imputed gene expression levels, imputed other omics are going to not be as accurate as if we measured the thing we're trying to do. And so to me, the fairly obvious solution to that is heterogeneous residual variance, additional on the amount of information that's within.
Well, I'm kind of wondering if it's a kind of problem that you think about the noise out at the end. But really what we're starting to deal with is noise in terms of the covariates that are going in there. And that's not the same thing.
No, you're right. And so I was realizing as you started to talk, also heterogeneous correlations. So actually imputed RNA seq's a different trait than measured RNAs. That gets us at least a little bit closer.
Yeah, Bob, I think the way Nancy put it is there's a few more PhD dissertations left out there to deal with these problems. Yes. Still doing good time-wise?
Quite over.
Okay. Any other questions? Or it seems like we've had a pretty good discussion. Yes.
They lots of research. Research costs money. The other question is, he says significant benefits to society. Should we not try to get a larger public years? There's an industry out there helped us in the past get money's into this kind of research. I think it's time that happens again. We're going to get this successful done. And sometimes it's going to cost a lot of money. The associations understand so much. The industry might put up and society that benefits. They should be getting paid.
I think that's a good point. I think I just want to make a comment. I think from the American Angus Association and kind of our research team, right, the way we've been trying to move along with the research piece, and of course funds is a big portion of that, but is to try to collaborate with universities, whether if we can participate with, we have the Angus Foundation that can sponsor some research, or, participate in collaborating, sharing the data for actually doing the research. So that helps sometimes to have an industry partner for a grant application or something like that. So that's how we've been trying to approach it, like not only doing our internal research with our internal team, but collaborate with different university groups, right? Whether that is through grants and different partnerships to promote that research component.
But the problem, and in class, it's going to take, I just find people without money. So that really is
I think kind of one of the challenges is getting young people interested in this area. It seems like it's looking at issues. It's always people is always the more challenging part, as opposed to the money you can usually figure out at some point. But if you don't have a person, the money doesn't matter.
So to Larry's point, I mean, we were fortunate at UNL for a while to have a couple of administrators and readers. Not that it was not the case anymore. But universities are in a lobby for what's best for their university, not to get common for it. So, and there's clearly money there. There's been a lot of money for grants, things like that, but not to tackle these kind of things. So I think industry needs a common voice, a common set of objectives to use for lobbying purposes to that end. We want to be put here, because without it, I don't see this.
AG2PI would be your most obvious first port of call, the agricultural genome-to-phenol initiative. There's a big pot of money, and that would be a great place to make a big project, open and across the spectrum.
There's that. Any other comments or questions? If not, I'd like to thank our panelists and also the wonderful discussion we've had.